Good afternoon, everyone. Thank you for joining us today for the Clinical Trials Methodology course webinar series. Today's webinar is on small sample size trials. Before I introduce our presenter, I want to mention that you can use the chat box if you have questions or comments. And you can unmute yourself by clicking on the audio icon to the right of the screen. Also, please remember to evaluate the webinar at the end of today's session. Our presenter today is Dr. Chris Coffey. Dr. Coffey is a professor of biostatistics and director of the Clinical Trials Statistical and Data Management Center in the University of Iowa College of Public Health. He received his PhD in biostatistics from the University of North Carolina at Chapel Hill in 1999 and has over 15 years of experience providing data management and statistical support to large randomized clinical trials. He is the principal investigator of the Data Coordinating Centers for the NIH-funded Network of Excellence in Neuroscience Clinical Trials and Childhood Adolescent Migraine Prevention Study, and the Statistics Core for the Michael J. Fox Foundation-funded Parkinson's Progression Markers Initiative. Dr. Coffey is a fellow of the Society for Clinical Trials, currently sits on the board of directors for the SCT, and serves on a number of data and safety monitoring boards. His research interests lie in the area of novel trial designs, particularly the use of adaptive designs. Thank you, Dr. Coffey, for presenting today.
All right, thanks, Lisa. So I think others are in the process of joining. Let me give it just a minute, and then I'll get started.
Very good. Just let me know when you want me to advance the slides.
I think I can share my screen. Let me try and see if that will work, and then I can just... Can you see my screen then? Okay, good. So I think, Lisa, the only thing is when I share my screen, I'm not able to see the chat box. So if you can look and if questions come up, just let me know and I can pause and try to address them.
Very well.
I can help you with that too, Chris.
Okay. All right, well, I think with that, why don't we get started? So what today's webinar covers is small sample size clinical trials, which I think will cover several things. So one of the points, and probably the most important point that I will stress, which I suspect has come up in a number of small groups, it's definitely come up in ours, is how do you overcome the challenges of small trials to address the importance of adequate study planning and verifying that your design is appropriate for addressing the question that you want to do. So if nothing else, I will hammer that point home multiple times through the webinar, that just because you're in a small clinical trial setting, it doesn't change the level of rigor that's needed to justify the design that you want to do. So what I'll try to do is really focus on that early on, and then talk about really two broad ways to try to address that in small trials. One that involves modifications to analytical approaches that you can use, and then I will very briefly kind of give a high-level overview of some designs that have been appropriate in the past for working with small clinical trials. This is the mandatory disclosures. I have really no disclosures of importance. The webinar is being recorded. Everyone knows that. I think Lisa mentioned the chat function that you can use if you have questions or if something comes up during the time. I will try to get through this with plenty of time remaining. So what I hope to do is to try to get through the slides and then allow plenty of time at the end to try to address questions. So if things come up or if there's something that's not clear, please use the chat function and I'll try to address those as much as possible towards the end of the webinar today. One of the things that is a challenge in small trials, so this slide is both, I think, a good example of setting the tone. Also, I'm originally from East Tennessee, so it's nice an excuse to put the hills of East Tennessee into the webinar. But if you look through this, I mean, one of the things in clinical trialists is when you look out over something like this, and with the hills, you know, from a trials perspective, you could see a bunch of bell-shaped curves, right? So it's this, what we would call the wonderful land of asymptopia, where, you know, in statistical properties, you know, with the central limit theorem and large samples, as you get large numbers, things all start to look normal. And a lot of statistical theory becomes much simpler when you have normally distributed outcomes or things that are asymptotically normal. In large phase 3 trials with thousands of participants, most asymptotic properties will kick in. Unfortunately, that's not always the case in small trials where we don't have these asymptotic properties that kick in. And there are really some subtle things that kick in, and there are some things that we take for granted, even in clinical trials, that break down in small trials. One thing I won't dwell on much today, but if any of you have had, have taken like a short course or taken a course on clinical trials, I'm sure you've talked about interim monitoring, where we do things like land-to-met spending functions or interim stopping boundaries to spend alpha to do interim looks. Those adjustments are all based on large sample properties. So even things like that are covered in basic texts of clinical trials only fit in when this asymptotic large sample properties are met. If you're in a small study with, say, 30 people and you're doing an interim look halfway through and you use the O'Brien-Fleming interim approach because that's what you've always been taught to do, it's not going to have the properties that you think it has because it's based on large sample asymptotic theory, which would break down. It would not be appropriate in a small sample setting. So one of the challenges of small sample clinical trials is a lot of the normally distributed aspects of statistical theory that we base much of the foundation of clinical trials on no longer holds. Now, one of the other things when you give a webinar like this is when you say what's a small clinical trial, that means different things to different people. And to many of you, it probably means different things compared to others on this webinar as well. So because the answer really depends upon the context. So in many instances, especially for common diseases, a small trial someone might think of as an early phase trial, which is what a lot of you are addressing in your proposals. However, in a rare disease, disease that can't be done in a large population, then a phase 3 clinical trial could be a small clinical trial. So I will kind of address both in this context. I put this in here to really try to make the point that there's really not a one-size-fits-all in a small clinical trial. We could be talking about an early phase trial. We could be talking about a very small rare disease phase 3 trial. I will focus more in the discussion here on kind of the early phase aspects because that is the part at which I think most of you are currently addressing as you try to work on your proposals. Now, the other part that's important is really no magic here. In some ways, these small trials are much more complicated to try to design than phase 3 trials because you're usually limited more in resources. The part that's really, I think, important and often lost in the discussion is just like, you know, your children are not little versions of their parents. They have their own personalities and their own characteristics. Small studies are not necessarily little versions of large studies. One of the things that's a common kind of red light with a lot of these early studies is if it's just a mini version of what you would ultimately want to do with phase 3 that's underpowered and has no ability to really answer a question, then you're not really gaining anything by doing the small study. We know that there are limitations. One of the things that you have to keep in mind is what are you sacrificing and with those limitations, and what are you willing to give up on one direction or the other? Less precision, a less definitive outcome. What are you willing to accept? What are you willing to give up? And how can you reach a compromise that still leads to a study that's meaningful and can address a question of interest at the end of the day? Now, before I get into our designs, there was this document. It's now about, I think, 15 years old. It came out in the early 2000s from the National Academy of Sciences on small clinical trials issues and challenges. One of the things that I think is most relevant is there were a number of key recommendations that came out of this. So the first, defining the research question, where it's just saying that with a small trial, it's particularly important that your question is well defined and that your outcomes and conditions to be evaluated are selected in a manner that will most likely help clinicians make therapeutic decisions. Seems pretty straightforward. Recommendation number 2 is to tailor the design. In other words, to choose a design that addresses the research question that you chose in recommendation 1. Recommendation 3 is clarify methods of reporting the results of clinical trials. So in particular with small trials, with limitations, it's important to clarify what characteristics and methods of data collection and analysis when you're doing the results, to make sure that all of the kind of strengths, weaknesses, and the compromises that I alluded to earlier that you had to make clearly come through. Recommendation 4, perform corroborative statistical analyses. That kind of speaks for itself. Obviously, recommendation 5, exercise caution and interpretation. There's going to be limitations because of the compromises. That's going to have to be taken into account. And then recommendation 6, which was kind of a catch-all, is that more research on alternative designs is needed. And really looked at, it basically said you can't necessarily take an off-the-shelf design that was done in one disease and plug it into another disease. There might be specific aspects of that, particularly if you're in a rare disease context or in something where you had the limitations of small trials with respect to limits of sample size. So here I've just pulled the 6 recommendations on here, and I say this is what came out of that document. The important point here is this is no different than any other trial, right? Large sample, small sample. The important aspects of making sure there is a question of interest, making sure the design is set up in a way to address that question of interest, and making sure that your methods are appropriate, your analysis reporting is appropriate, your interpretation of the results is appropriate, and trying to come up with a design, perhaps some alternative design that's more innovative compared to what has been done in the past that might be needed in order to address that question. So this really reiterates what I mentioned at the beginning of today's webinar, that it's really no different from any other trial. The important aspects of how do you come up with a question, how do you make sure you have the ability to address that question, is something that's equally important in a small sample setting, just as it is in a large sample. And there are really 3 basic requirements in order to do that for any clinical trial. As I mentioned, it should examine an important research question. It should use rigorous methodology to answer that question. And it needs to be based on ethical consideration and ensure that the risks to subjects are minimized. Now, you may have to compromise somehow on some of these or make modifications in order to address that. What you can't really do, and in particular, if you're writing a grant I can give from my experience reviewing grants for foundations or for NIH study section, if a trial comes through that's limited with respect to sample size, limited in terms of the feasible number of subjects you can report, there might be a clinically very important question. But if the methodology is not such that you can't really get an answer to the question, then it's not really of interest from a funding perspective. So if, for instance, one that comes up a lot is people might have a primary objective to show safety, right? Safety is a horrible objective in a clinical trial. Not that we shouldn't be looking at safety of clinical trials, but if you're trying to show that a drug is safe, you're really shooting at a target that you're never going to achieve. We can really never show that a drug is safe, even after doing large phase 3. That's why we have post-marketing evaluation of drugs, even after FDA approval. We can only show that it's safe to a certain extent. And one of the key aspects of those early safety trials is understanding to what extent are we able to kind of rule out that the drug is unsafe at that perspective. In a very small early phase 1 study, you're probably only going to see really, really major adverse events or toxic events of interest that might limit moving forward. That doesn't mean that not meeting that implies the drug is safe. The drug could very well be unsafe. It's just unsafe to a level that you weren't necessarily powered to detect in the early phase. So it's important to keep into account what the question is, what you can address. A useful exercise is often to come through and like at the end, if you have a question, how confident are you that your result is correct in terms of what that would imply to moving forward? Or if you find that it doesn't meet the criteria you had, what would you conclude? Which if it's not convincing to you that your drug, your intervention didn't work, then you should also think about how could the design be tweaked so that at the end of the day, we have an answer to the question one way or the other. One of the important points that I often make when I talk classes and trials or talk to collaborators is there's a difference in terms of what I think from a design perspective should be considered a negative trial and what is often considered in the media as a negative trial. So a trial that assesses a drug that doesn't show benefit on the efficacious parameter would be viewed as negative when it's reported, in the newspaper or the TV, the internet. But as long as it addressed the question of interest, right? If the drug didn't work and it showed that it didn't work, that's a successful trial. The question of interest was addressed. So a negative trial from a design perspective would, in essence, be one that completes, uses up all the resources, but doesn't address the question. So you end up with a study that's not statistically significant at the end of the study, but it's not clear that that's because the drug didn't work or because the study was underpowered, and you really don't have any more information at the end of the study than you had at the beginning of the study. That would be a negative trial in terms of the design perspective, and that's what a lot of the discussions you think that you're probably having with your small group leaders at this point is trying to address. How can you make sure at the end of the day you've answered your question of interest one way or the other, and how can you be confident that whatever answer you've got is likely to be correct? Now, there are different approaches in terms of how you could relax those criteria in terms of addressing those. So what is really important and most critical is trying to do that in a prospective way instead of a retrospective way. So really trying to make a case from the beginning, deciding on what you're willing to accept, what you're willing to maybe live with a little bit, because that becomes really important for how you would report the results and how you would interpret the results and how that would influence what your next steps might be. So if you know you could only roll out large safety differences, you're going to have to account for the fact that in your next study, there's likely to be some risk that you would want to account for. You can't conclude that the drug or the dose that you've chosen is completely safe because you've only ruled out perhaps large risks. I mentioned early on that there were two general approaches you can use here to try to help your ability to address these. One is analytical and one is from a design, and I'll just briefly talk about each of those. So from an analytical perspective, one of the things is it becomes really critical to choose outcome measures that are as efficient as possible and precise as possible. And so I've got this is the one formula I think they have in the slides today. Statistician has to have the formula on the slide. But in general, the detectable effect is related to the ratio of the variance to the sample size, right? And so as you know, if you've gone through a sample size calculation in any type of setting, whether it's clinical trials or not, as the sample size increases, the variability will decrease, right? So in a large setting, we can always get more subjects in order to reduce the variability around the effect we want, which will increase our power. And that's a lot of what we end up when we go through sample size calculations that we do. And in some ways, if there's a little, if the variance is slightly larger, we can take a large enough sample to overcome that. If we're limited in terms of the sample size that we can do, then the only thing that we could really try to do in order to gain power would be to increase the precision of the variance. And so really understanding the outcome measures, understanding the properties, and putting techniques in place to make sure that we get measures that are as precise as possible would be really critical in terms of trying to address those concerns from an analytical perspective. So in terms of the type of outcome measure, this is something that's not really considered in a lot of situations as much as it should. But there are different types of outcome measures that often exhibit different levels of accuracy. And they also then in some ways lead to that accuracy translates into differences in statistical power. A thing that's very common in clinical trials is to use binary outcomes, where you might group some type of continuous measure into a dichotomous. So I've got examples on the next page. So ALS might have a binary of a 10% decrease in the ALS-FRS-R score or not, or for pain, a binary of whether the pain score was greater than 4 or not. You see these commonly in clinical trials, particularly in phase 3 trials. They're easier to interpret, they're easier to kind of summarize, but they're less efficient. Continuous outcomes tend to be more efficient, and time to event outcomes are somewhere in between. So what that really deems is, unlike we may often do in phase 3 studies in a small setting or in a rare disease setting, if there's a continuous outcome, if there are ways to model the continuous outcome itself, that can be more efficient than trying to dichotomize that continuous outcome in some way to address the question. And on the slide here, you can see the examples that I mentioned before from ALS, how you would cast each of those in different ways. So you could have a binary endpoint of whether there's a 10% decrease or not, or you could have a time to event of the time at which you reach the 10% decrease, or you could just look at a pure continuous of modeling the ALS FRS score over time and looking at the change as a continuous variable. And similar things for pain score. These are types of discussions that should take place early in the design. In the phase 3 and the large sample setting, it often is kind of a minuscule difference because we can choose sample size for each, although it affects the budget, and that's always something that we consider. But in the small sample setting, it might be critical. It may be that you can't get the sample size you would need to have adequate power with a binary endpoint, but perhaps you could with a continuous endpoint, because that extra bit of efficiency may be critical in terms of assuring what you can do. Same thing for parametric versus non-parametric approaches. So non-parametric approaches don't require assumptions. Parametric approaches do. And one of the advantages of parametric approaches is that they can lead to higher power if those results are satisfied. The disadvantage to parametric approaches is that they can lead to erroneous conclusions if those assumptions that go into the distributional results are not satisfied. So using parametric approaches can typically gain you power in small sample settings, but it's very, very important to know whether the distributional assumptions are satisfied. As an example, going back to what I alluded to at the very beginning, if your analysis methods assume normality, you would need to have some level of confidence that even in the small sample setting, you have normally distributed endpoints. If you do not, if there was some skewness or some other deviation from normality in your data, you could end up with biased results because it would not satisfy the distributional properties that you're applying to it. So that's an important thing to think about. A part of this, and this kind of goes back to another point that I made at the beginning, the design of early phase or small trials can in many ways be more complicated than the design of large phase 3 trials that are trying to, from a confirmatory perspective, because you're often going to need lots of conversations and a real collaboration between the clinician and the statistician to address many of these issues and to make sure that you're squeezing out as much efficiency as possible. One of the roles that the statistician often plays in these types of studies is the person who kind of has to bring it back to reality, and that if it's not feasible to address the question with the limitations of the sample size available, then it's not necessarily going to be the best use of the team's time to try to flesh out a protocol and a grant that's not going to go anywhere, because the study section is not going to fund something if it's not capable of answering a question of interest. And one of the things that I was joked around once, but there's a lot of truth to it, is that someone once referred to statisticians as the killjoy in a room of clinicians, and that's one of our roles, is the practicality aspect of how feasible is this to do? Can we address this? And to kind of illustrate it slightly different in terms of how to increase power. So in a type 3 setting, you can be model free as possible. You can do large sample size. You do intent to treat analyses. You worry about noise to an extent, but you don't overly obsess on it. And really, doing phase 3 trials are very much like this photo you see here, where the little kids are all running in a race. If one kid falls down, there's so many kids, you might notice it, but it's not going to draw a lot of attention. Where small trials are more like here, where you're the lone runner coming across the finish line, or if you trip and fall there, everybody's going to see you at that point. And that really addresses the challenges that it's much more, everything is much more critical. Every decision you make in terms of trying to increase power is much more critical in the small clinical trial setting. So for instance, we mentioned the parametric models. You should use those as much as possible, but they should be pre-specified, and you should make sure that the assumptions are met. Really checking each observation. Data cleaning is critical in any clinical trial. It's extremely critical in a small sample clinical trial. If you have lots of sloppiness in your data, it can offset any of the advantages you have. And then the latter is especially important. Carefully evaluating alternate designs is really critical in small trials, because it's most likely that you're going to have to try to assess the design in such a way and come up with a design that is the absolute most efficient for the question that you're looking at. A couple of things that come up, and these have come up in some of at least the small group discussions I've been involved with, and I suspect others. Historical controls can be a good way to try to minimize the numbers, because you then no longer need a placebo group. there's lots of interest in historical controls from a number of groups, particularly patient groups like this, because a lot of people would rather join a clinical trial if they don't have a chance of getting placebo, if they know they're going to get the active treatment, even though there might not be any benefit or any guarantee that that active treatment would work. So historical controls are important in those situations if the data from published studies remains relevant. One of the holdups with historical controls is that you really, really have to have a solid understanding and a lot of confidence that what you know in the past is going to be predictive of what your control group might look like in the study. Unfortunately, there's a number of examples that have shown problems with historical controls. Two major issues. One, there are temporal trends over time in diseases. Sometimes that might not be the case, but even when that is the case, there's a huge literature that has shown numerous times that individuals who participate in clinical trials tend to have better outcomes than the population as a whole. That's whether or not they get treatment. There are huge placebo effects. So even subjects on placebo tend to have better outcomes than you would generally see in historical controls or in registry data over time. So if you use historical controls and your control group did you have one, is going to look better than historical controls, that may lead to erroneous conclusions in terms of treatment effect and would make it hard to determine whether you actually have a true treatment effect or not. And that's one of the big concerns with historical controls. So they are a tool that can be used. They help. They can reduce the cost. They can often reduce recruitment, and they can enable you to do that in a more efficient manner. But you really have to make a strong case that the historical controls are expected to be highly representative of what you would have gotten had you enroll a placebo case, which is in many cases a high bar to cross unless you have really, really convincing long-term registry data, which unfortunately doesn't exist for many diseases. There has been a lot of attention in recent years to trying to develop registries for a number of different diseases to try to develop more of that information. And I suspect over the next decade or so, we'll see more of those types of efforts put in place. So outside of the analytical approaches, other types of designs of interest that can be used in small clinical trials, I'll talk just very briefly about 5 here: repeated measures, crossover, N-of-1, futility, and ranking or selection designs. So repeated measures design, this is pretty straightforward. We do this often in clinical trials where you'll take multiple observations on your outcome for each subject. So repeated measurements over time, which would be longitudinal data, or perhaps multiple measurements on the same subject where you have multiple outcomes. Now, one of the things that you often see in phase 3 trials is you might have outcome data collected quarterly, twice a year, yearly over a long period, but your primary outcome might be the change from baseline to the end of the study. So again, you're just looking at that as opposed to modeling all of the longitudinal data that you have. One of the things with the longitudinal data is with the between subject and within subject comparisons you could do by having measurements over time, you can explain more of the variability from a subject because you can use them as their own control. So instead of measuring the baseline value difference from final value, if you can model the longitudinal data as a whole, modeling the variance covariance structure, you can really explain much more of the variability and increase the efficiency of the clinical trial. That's critically important when you're limited in terms of sample size in small trials. And so if you have longitudinal data in a small trial, as much as possible, you would want to try to model that data over time and use those longitudinal models to then assess your primary outcome of whether you're seeing any difference, even if it's over a 2 or 3 year period, because you're going to explain more of the variability. And if you explain more of the variability, then you're reducing the variance of your outcome measure, which is going to increase power and could be a very important way to address it. There are crossover designs. Crossover designs come up a lot in limitations here as well, where I know many of you have had these discussions as well in small groups, where the advantage of a crossover design, that there's a recruitment advantage, just like there was with historical controls, because everyone knows they will get the treatment at some point. With a crossover design, you're randomizing the order of treatment. So some people may get drug first and then placebo. Others might get placebo and then drug. But ideally, you have each individual serving as their own control. And so you can then reduce the variance in that way, which again increases power, which is very attractive from a small sample size setting. The disadvantages are that you really need to have a long-term disease with outcomes that can occur and be measured over time. But then the individual kind of is back to where they were at the beginning of treatment once the treatment is ceased. So if you had a situation where, say, you're looking at two individuals, if you treat the one with drug first and you treat the second person with placebo over, say, a 6-week period, you collect your outcome, they wash out for 4 weeks, and now you flip, so the person who got treatment gets placebo, the person who got placebo gets treatment. If they're not at the same place with respect to the outcome, so if the person who got treatment has some lingering benefit of treatment that the person who got placebo would not, then there is what is termed a carryover effect. And a carryover effect can be problematic because that means that the order at which you receive the treatment makes a difference. And so you now just can't use an individual as their internal control, because getting treatment after placebo is different versus getting treatment before placebo. And so if there's carryover effects, it really it limits a lot of the advantages that you get from crossover designs and causes a lot of issues. And so that's something that has to be carefully considered when planning a crossover design, is assessing what is the extent of potential carryover effect and trying to decide whether there are systems in place that can address that. Another design that is often useful, particularly in extremely rare diseases, and this is very similar to the crossover design in some ways, is what's known as an N of 1 design. So an N of 1 design would be a special case of a crossover or repeated measure design where a single subject overgoes treatment for several pairs of periods. So it's very similar in the crossover design in that each pair, it's randomized whether you get treatment first then placebo or placebo first and then treatment, but you might do multiple pairs for the same individual. And you would see this, again, you have the carryover issue that you have to account for just as you do in crossover design. But say you had a very rare disease where you have exacerbations and you're trying to treat the exacerbation. And so you have an exacerbation, you treat it, it goes away. You might have another exacerbation, you treat it, it goes away. That would be the setting where an N of 1 design would be extremely helpful. Even in something that's more common, this would be useful for something where what you're treating is more acute. So for asthma attacks in an asthmatic subject. And the idea being, if it's a very rare and you have these exacerbations, you could treat the individual at each exacerbation in a way that's randomizing, in the pair, which one do you get first? And you get multiple pairs over time, which will enable you to explain a lot of the variability, look at the within subject changes, and try to get a sense of whether the individual is doing better on the periods of exacerbation when they were on treatment than they were during the periods of exacerbation where they were off treatment. And so where it's maybe not feasible to do many subjects at all, these N of 1 designs can be extremely useful. It's also, if you do these on multiple individuals, you can often combine a series of N of 1 trials using meta-analysis techniques the same way you would combine data from different studies. And so if it's only feasible to do very few subjects, perhaps a few N of 1 trials could then be combined to try to get a little more information across multiple subjects with the disease. So this is something that is of interest, particularly with an extremely rare disease where it may not even be feasible to get perhaps 50 subjects enrolled in a clinical trial. There are selection designs, which are another helpful thing in early phase studies. This would come in a situation where you have perhaps multiple diseases, or in some cases even multiple drugs to choose from, and it wouldn't be feasible to do a very large study of each drug or of each dose. And so it's kind of a quick way to try to choose the one that looks most promising, which in essence, a selection design you could think of as a horse race, where you put multiple drugs or multiple doses of a drug, you treat individuals, you randomize them to different drugs or different doses of a drug, and you look at the response and you select the best treatment out of the K potential treatments and use that as a screen to determine which to look. So selection designs would be useful. Let's say you're in a disease area where there are a number of different treatments that are out there that have been heuristically, there's some evidence to think that it might be helpful to treat the disease. Say there's 12 of those. In order to screen all 12 of those in a highly rigorous trial would be costly, and if you did them separately, it's going to take a long time, and it'd be unclear which to choose first. You could put all 12 in a selection design, treat some number of subjects just to see which one has the best response and use that as a screening to determine which one you should then invest more resources in a later trial to try to get a better sense of, in some cases, proof of concept, and then perhaps a confirmatory phase 3. It has a high type 1 error because you're not really doing any testing. You're just looking at which is the most promising. But it is an interesting thing to screen through a number of treatments, particularly if you have, as you see at the bottom of the slide, what's often known as the pipeline problem, where there's too many treatments to study. You need to come up with some way to try to determine how to prioritize the resources in looking at those various treatments. The final design I'll talk about is what's called a futility design, which is very useful, but a lot of people don't like it at first because it's kind of got a horrible name because most people don't want to show futility. But the idea of a futility study, really, this is more of a phase 2 type of design, where one of the universal problems of phase 2 is there's often uncertainty in terms of how to design phase 2, because if you're using the endpoints that are similar to what you would use in phase 3, then you're either going to power it, in essence you're doing the phase 3, or it's underpowered, in which case it's an underpowered phase 3, neither of which are optimal. The futility design is kind of an intermediate compromise, which enables you to screen at phase 2 to determine whether or not it looks promising enough to go to phase 3. So with the futility design, you're kind of flipping the null and the alternative from what you would typically do. So instead of trying to show that the treatment is better, you're trying to set it up so that if you reject the null, you would declare that it's worse to the extent that you wouldn't see the effect of interest in a future phase 3 trial. So if futility is declared, you would reject the null hypothesis and declare that it's not cost effective to conduct a phase 3 trial. If futility is not declared, then you do not reject the null hypothesis. However, if you remember from your basic stats class, you can't prove a null hypothesis. You can reject it, but you can't prove it. So not rejecting a futility suggests there could be a clinically meaningful effect, or there could be no difference. It could be a difference that's much smaller, but not of clinical interest. But you don't have enough evidence to declare you're far enough away from the effect to declare futility, which would suggest it's probably worthwhile to expand into a larger phase 3 trial to really try to get a better sense of efficacy. So for instance, let's say you're testing some treatment and a 10% increase in favorable response rates over placebo is clinically meaningful. In a futility design, your null hypothesis would be that your treatment improves outcome by at least 10% or more compared to placebo. So you have the effect or greater that you would power the phase 3 study if you were to do it in the future to detect. Your alternative hypothesis would be that the treatment does not improve outcome by at least 10% compared to placebo. So if you reject the null hypothesis, you're far enough away from that 10% or better improvement on treatment that would tell you it's futile to consider in a phase 3 trial. It's not likely that you're going to see that clinically meaningful effect. I have this slide here that I won't dwell on, but it just kind of shows the statistical properties of how the null and the alternative are kind of flipped from what you would see in a usual design. Now, because of the way it's set up, the futility design has high negative predictive value. So if you declare futility, it is very likely that the treatment is not effective. And that's a good thing to have. That's what we want to do in screening. It has low positive predictive values, however, because this gets back to the point of you can't prove the null hypothesis. So lack of futility doesn't imply that the treatment is effective. That's in essence, going back to the very beginning of the webinar today, that's what we're giving up here in order to address the limitations that we have in this setting, is that just because we don't declare futility, we can't conclude that we have an effective treatment. We would need a larger future study in order to further explore that to determine whether we have an effective treatment or whatever benefit is there is not far enough to be away from the 10% to declare futility, but not high enough to really be meaningful. And this is a very big improvement over running underpowered phase 3 trials in phase 2, which is a way to have some rigor of screening, which is a compromise in order to have a question of interest that can be addressed rigorously in order to determine whether it's meaningful or not. And in and of itself, it can actually be a very powerful thing. So there have been a number of groups that have looked at failed phase 3 trials and kind of done some retrospective looks to look at had a futility design been done in phase 2, it might have prevented doing the phase 3. And as much as from investigators, we want all the treatments that we're looking at in trials to work. That's one of the reasons why all of you are investing your time working on these protocols. unfortunately, the truth of the matter, in neurology, is that more of you are going to fail than succeed. Right? There are many more trials that fail to show treatment benefit than are successful in showing treatment benefit, particularly in the neurological sciences. And so, because of that, if the designs can be set up so that if something is not going to be promising, you could detect that earlier, that's better for the field as a whole. It's frustrating for the researcher who has poured several years into looking at that treatment, but it's much better to fail early. If you're a company, it's way better to fail early because you're using up less resources, less investment, less investment has gone into that. And so a lot of these designs are really what we're trying to do here, structure it in such a way that if it's not going to be meaningful to go forward, if it's likely to fail at phase 3, we would rather it fail in phase 1 or phase 2 so that we can determine that without putting as many resources into it. The last thing I'll mention, which is more of a global thing, is something, so in looking at alternative designs, there has been a lot of interest in what is known as adaptive designs, which there will be a little discussion of this at the course. But adaptive designs are really an innovative trial design as a whole, are something that has received attention where it allows reviewing information during an ongoing trial. So one of the limitations in kind of the standard approach is that you have to make a lot of assumptions, right, in order to set up the design. We talked about that earlier even in this webinar. If those assumptions are wrong, then your design is completely wrong and can lead to an erroneous question. Adaptive designs try to account for that uncertainty and to give you avenues to make changes to account for that. There are two general definitions, one that came from the Adaptive Design Scientific Working Group, which is currently under DIA, who published a white paper in 2006. And then there's also a draft guidance from FDA in 2010. Both of these define adaptive design in an important way for someone who wants to consider that. And that adaptive design doesn't mean adaptive kind of in the sense of how Webster's dictionary would define adaptive. And what adaptive designs from a regulatory perspective would be a study that has prospectively planned opportunity for modification. So it's really pre-planned adaptation. So you kind of know what uncertainty you have at the beginning. You're specifying how you will address that uncertainty kind of in advance by the design. And what I mean by that is you're kind of specifying at some interim point, if this occurs, we'll do A. If this occurs, we'll do B. If this occurs, we'll do C. That's critically important in order to accomplish the first objective under the Gallo thing without undermining the validity and integrity of the trial. So if we've specified what we would do, we can conduct simulation studies under a number of different conditions to look at A, whether there is any bias introduced from the adaptation, which is always a concern. If there is, do we have proper techniques in place to account for that bias, right? So one of the concerns with making adaptive decisions based on ongoing data is it can become data-driven, which any type of data-driven analysis is prone to bias. And so that's something we have to be constantly concerned about. If we specified it in advance, we can assess the bias through simulation studies. If we didn't assess these in advance, let's say we started doing a study, we get halfway through and we realize our assumptions are erroneous and we want to make changes. At that point, we can look at the properties of the study conditional upon making that decision and moving forward, but we can't rule out bias from the study as a whole because we can't say what we would have done had something different happened at the intermediate. And so there's always the potential for bias, which is why from the regulatory perspective, it's critically important to have all of your design modifications, even from the adaptive aspect, prospectively planned and vetted ahead of time. So in general, with adaptive design, all groups from kind of the regulatory perspective and most of the discussions of adaptive designs now focus on this concept of adaptive by design, and that only planned adaptations can be guaranteed to avoid any unknown bias, and as much as possible, if there are going to be adaptive components in the clinical trial, they should be planned in advance and part of the design itself. So this is, this has been a lot condensed in here, which is like I think most of these webinars, where you can almost talk for a full day or longer, maybe even week, about the designs in general. To kind of summarize and to kind of hit the key points, one of the things that you should definitely take from this is appropriate study design, really no matter where you are, phase 1, phase 2, 3, whatever disease you're looking at, should have sufficient sample size, adequate power, control of bias to allow a meaningful interpretation of the results and to address some question of importance at which the trial is set out to do. And although that's more complicated in small clinical trials, you can't ignore those issues. And that's really the most complicated thing in trying to work with phase 2 trials is how do you balance between both of those? The limitations that you have just because of the rareness of the disease or the limitations of sample size for what you can do versus how can you make sure that you're still addressing those properties in a way that will have validity at the end of the study. So the majority of methods are large sample theory. That's something that you have to keep in mind. Additional research into innovative designs is clearly needed. But more so, it's really important, as I think you're probably seeing as you've gone through your small groups, that there's a discussion of teams here. You really need the clinician and the statistician working very closely together all through the design phase in order to come up with something that works appropriately. And with that, I will pause and take questions.
I think you need to put up the link for the surveys again, Chris.
Oh, did I forget? All right.
Or let Lisa share her screen and do it.
Okay. Lisa, can you share? For some reason, it won't come back up on that. We can email the link too if that's an issue. Seems like there's one question. With respect to parametric tests, I was under the impression that once you get to approximately 30 participants per arm, central limit theorem relaxes requirement for normality for parametric tests. Is this reasonable, accurate as a rule of thumb? In many situations, yes, that's something that I think many people are taught. It's not universally true, and it kind of depends upon how far you are from the assumption of normality at the, from what you start. So it's kind of a standard rule of thumb that most people are taught and holds reasonably well, but it doesn't, you can't necessarily take it as kind of gospel and assume that's always going to be the case. It's a good question. Any other questions?
Chris, this came up in our small group after you left, talking about crossover designs with small numbers and the washout period. Is there ever a utility in using a placebo during the washout period to prevent people from using other medications? Or actually, as I'm asking the question, I'm thinking I'm answering it already in my head, but
I'm not sure how that would work.
Yeah, I don't think it would work, never, because the problem is that everybody you'd either have to blind them to when they were in washout. Never mind. Once I said it out loud, I knew I was confusing myself.
Yeah, I mean, crossover designs or something, it comes up a lot. People always want to use those because you can limit your sample size. I would say, I mean, there are situations where they can be really useful. I would say probably 9 out of 10 conversations I have with someone who wants to use a crossover, when you get deep into this carryover issue, it becomes unclear whether the assumptions can be met to really gain the advantages of it. So it's something that has to be carefully evaluated and thought through if you're considering a crossover design. And it's something that will absolutely come up in review. The question, is the following a recipe for disaster in small trial sizes, placebo versus treatment 1 versus treatment 2? That's a tricky question. I wouldn't necessarily say it's a recipe for disaster. It depends upon what your questions are. And a lot of the issues that I talked about are going to be even more compounded. For instance, say you're comparing treatment 1 versus placebo and treatment 2 versus placebo, because now you've got multiple comparisons, you've got 3 groups. Depending upon how limited you are in sample size, it may be more difficult to address those. But if you can still address them, I think it can be successful. I think the challenge is, so I wouldn't call it a recipe for disaster. I think it's going to be more challenging. Another question. I'd not thought of N of 1 trials much. Do you have thoughts about when to use crossover with 2 times process versus multiple N of 1 with meta-analysis, say for a trial of size 20 to 30 patients? Well, I think so. Really, a crossover design with multiple crosses is kind of very similar to an N of 1 trial. It's really a combination of different N of 1. That's why I mentioned you could combine N of 1 trials using meta-analytic techniques. If you can do the crossover and if the assumptions are valid with 20 to 30 patients, I think I would prefer to stick with the crossover design. A lot of the N of 1 typically comes up mostly in like extremely, extremely rare diseases where you may only have access to like one person at a time. Anything else? Right, another question just came in. Mortality is my outcome of interest, and by definition is categorical, but in this case, I assume time to event would be the way to go. Am I correct? And in that case, how would you estimate how long you want to follow over time? So, yeah, I mean, you're going to get more power by looking at time to event, because then, and then will also, it can give you not just more power, but better ability to discern, because depending on, if you look, say, over 5 years, which I don't know what time frame you're talking about, over 5 years, you might see equal mortality rates on drug and placebo, but if participants on drug are living longer within that 5-year period, that's an important thing to keep in mind. So you would lose that by dichotomizing, mortality at 5 years. So you can look at, not just are the percentages. So it depends on what your disease. I have no idea of what mortality rate you would expect on that, but you can get a sense of, are you shortening the time to mortality as well as mortality as a whole. In terms of how long you want to follow over time, that's something that would be one of the design decisions that would have to come up, because it's going to affect your power. It's something that you probably want to discuss with your team leaders and statisticians. There's no right or wrong answer. It's going to be very study specific. Doesn't look like anything else is coming in. Let me just take one last opportunity to thank everyone for joining today. Hopefully your protocols are coming along well, and we look forward to seeing everyone in Ann Arbor in a few weeks. Thanks a lot.